Welcome everyone, good morning. We're gonna give this a start because it's 8:01, so just to make sure that we have enough time. We're lucky to have two speakers today. We have Dr. Katie Riggs and Dr. Scott Letendre. And so we'll get going. I'll just give you a little bit of background information about Dr. Riggs, who joined the faculty at the University of California, San Diego in August 2024 as an assistant professor in the Department of Medicine, Division of Infectious Diseases and Global Public Health, following her postdoctoral research training on the HIV and other pandemics, the HOPE T32 training grant at UCSD. Her primary research focus is on how viral co-infections shape host immune responses and contribute to neuropsychiatric complications, particularly in people with HIV. Supported primarily by a KL2 through the ACTRI and CIFAR Developmental Award. She balances her research with continued ethical commitment to her patients at Owen Clinic and on the inpatient ID consult services. I think I'll introduce Dr. Letendre once it's his turn to speak, just so that it's fresher in your mind. So please, Katie, take it away.
Okay. Hi, everybody. Thanks for joining. So as we said, I will do part one, and then Dr. Letendre will take over the second half. And so it probably makes the most sense to just ask questions kind of as we go along. So we'll get started. So no disclosures. So I just have kind of two little sections for me. So just talking, there was a lot of stuff at CROI this year about pathogenesis and biomarkers, but I'm just going to highlight a couple of them. So for the first one, this is in older adults. And there's a big question and a big emphasis this year as well on how do you distinguish in people with HIV who are aging, how do you distinguish from kind of age-related or Alzheimer's type dementia from HIV-associated cognitive issues. And so what they've done is this group, this is all from the HIV NAT 207 or TI aging study. And they had 255 people with HIV enrolled. They were all over 50 and all virally suppressed. And they wanted to know if a plasma biomarker of Alzheimer's disease, this is phosphorylated tau 217, would predict cognitive decline in older people with HIV. And what they report to note that they have basically 2 visits, most of them are approximately 6 years apart, and they have MoCA values. That's their only neurocognitive assessment, but that's at baseline and at 6 years. And then they just had the biomarkers at baseline. And so they did find that higher baseline phosphorylated tau 217 predicted cognitive decline. And not too surprisingly, there was an interaction with age. So this decline or this relationship between phosphorylated tau 217 and cognitive decline was much stronger as people aged. And so this is the graph here is basically the red line is the top quartile of p-tau at baseline, and then the cognitive trajectory, so total MoCA score. And it was I think it's a little bit surprising. They did find, pretty significant difference over 6 years for these individuals, or I guess this is the interaction effect, but there was about a decrease in the top quartile. There was a decrease of MoCA scores over 6 years by about 4.3 points. So that's definitely in the significantly impaired range for MoCA. And so I think kind of the takeaway for me from this is that, this doesn't really necessarily have any controls. It doesn't speak to whether this is a different relationship in people with HIV, but I would say it may or may not be. But it sounds like this Alzheimer's disease diagnostic biomarker, which is a plasma biomarker, which is great, does show promise in predicting cognitive decline in aging people with HIV. And that is something we can do in clinic, and that is a reasonable screening to get started on diagnosis of Alzheimer's disease. And then this is a very different cohort looking at markers of neuronal injury, and this is jumping into CSF samples. And this is, they wanted to see if you treat people during acute infection with ART, does that prevent some of the neuronal injury? Or does it reverse? And so they measured several different markers of neuronal injury in this acute cohort. Note that the mean age is about 27 years old, so very young individuals overall. They measured these markers at diagnosis and then at post-ART follow-up. And overall, they do have a group of 44 people without HIV that were demographically matched overall. And so what they found was that, yes, at week 0, so at acute infection presenting to the clinic, there was elevated NFL in the CSF, so a marker of neuronal injury, and this was higher than people without HIV. And that this did not, or this remains significantly different from people without HIV, even at week, this shows up to week 48, but also they had up to 5 years, 240. And I will say, so and it did, at week 0, the CSF NFL correlated with the CSF HIV RNA level, which is not surprising. And it did negatively correlated with CD4 CD8 ratio. But I think the big takeaway is everybody wanted to see this a little higher at week 0 and then significantly decline and normalize after initiation of ART, which was not what was found. But I think it is important if they look at it binary of, above upper limit of normal for NFL elevation, that it was 15% of people at week 0 had elevated NFL. And so it's not across the board. And that even though there wasn't a statistically significant decrease on follow-up, the percentage of elevated NFL at follow-up was 5 to 9%. So still a relatively minority of individuals, and this does not have necessarily clear correlation with, at least in this study, with cognitive symptoms or any neurosymptoms. And then, their conclusion was that early ART alone is likely insufficient to provide complete neuroprotection in all people with HIV, which I think is a reasonable conclusion and something we kind of already knew, but it obviously goes a long way to help. And then this is more biomarker evaluation, but also looking at trying to see the relationship of polypharmacy. And polypharmacy was, like last year, a definite theme, and there were multiple different posters and some talks on polypharmacy. This one was looking to see if, or they were trying to investigate and get a sense of whether kind of polypharmacy-associated neurological disorders are mediated by neuronal damage, inflammation, or if it's simply a reflection of high comorbidity burden. And so they looked at polypharmacy and falls, and they measured 3 different measures, neopterin, and this is all in CSF. So this is CSF neopterin, kind of a general immune or inflammatory marker, soluble CD14, a primarily monocyte marker, and then NFL, which is what we talked about before, which is kind of a marker of neuronal injury. And so even after accounting for comorbidity burden, age, and sex, they did find that there's a significant association with increased risk of falls and CD14 and NFL. Interestingly, didn't find a relationship with any of the markers on univariate or multivariate with slow gait speed, but it was just with increased risk of falls. And then for multivariate analysis for just polypharmacy, these were also associated with the CD14 and NFL, but not neopterin in CSF. And so for this, in this, you don't have to necessarily read this, but basically it just shows this bottom panel here shows this is comorbidity burden. So all three of the measures kind of gradually increase with increased comorbidity burden, which is not particularly surprising, but interesting to see, and a reason that they did have to control for comorbidities. And so this is just restating that they did find that polypharmacy and recurrent falls were associated with NFL and CD14, even after adjusting for comorbidities. The next statement was that polypharmacy contributes to neurologic disorders in older PLUH, and medication management may improve brain health and geriatric syndromes. So yes, I think that this is something we all definitely keep in our minds. I'm not sure this study proves definitive causality, but it is a good reminder. And then just two kind of quick notes of co-infection related posters. So this was an interesting study that looked at, this was done in 811 participants with cryptococcal meningitis in a multi-site trial throughout Eastern Africa. And this was looking at the liposomal amphotericin treatment, but this is a prospective nested cohort. And they measured CMV viremia, or CMV and EBV in blood and CSF. And what they found was definitely differential effects for prognostication in CMV viremia and EBV. So for overall, about half of the people had CMV viremia in blood, and about 5% had it detectable in CSF, and 73% had it EBV detectable in blood. And they found that CMV viremia with a level of greater than 1,000 was associated with more of a POSI immune response, which we know is typically associated with worse clinical outcomes in cryptococcal meningitis. And that's what they found here as well. And so this POSI immune inflammatory response was lower CSF white blood cell count, so this is less than 5 for median. And that was significantly different from the people who did not have any detectable CMV. And interestingly, the CSF fungal burden, which was significantly much higher in people with CSF or detectable CMV viremia. As you can see in this, I'm sorry, using the wrong screen for my pointer there. So as you can see in this box here, the CSF fungal burden was much higher. And they did find that most notably there was a 2-week mortality adjusted odds ratio of 2.3 for people who had detectable CMV viremia above 1,000. And interestingly, EBV viremia was actually, they said, was associated with more of a pro-inflammatory phenotype, but I would say it was statistically different, but overall, they didn't see that EBV had any increased mortality risk with it, and in some ways, it almost looked more favorable. So they do say, and this is in quotes again, they say that CMV viremia represents a potentially modifiable risk factor to improve survival. And they are, at least according to the author, as they've already started looking into preemptive CMV treatment trials in people with cryptococcal meningitis. I guess, again, I'm not entirely sure. I think we've had the debate many times of if someone is already much sicker, they're more likely to have CMV reactivation or viremia. So there's not a clear causality, at least to me, in this, but I think it is certainly is shows that it is a strong prognostic marker, and I'll be
Katie,
interested to see. Yes, hi.
just one point. I mean, Sarah presented data from the Termavir that, over a relatively short period of time, there was like improvement in the CD4, CD8 ratio, a totally different population, but there is reason to think that suppressing CMV viremia could improve immune responses.
Oh, I absolutely. There,
Thanks.
absolutely.
That'd be pretty interesting.
And
Thanks.
Scott's going to talk about it.
Thanks for ruining my presentation, Tim.
No, you have all the pretty pictures, Scott. You can show the pretty pictures.
No,
But yes,
yes.
excellent segue and foreshadowing. But I would agree. I'm interested to see. And so, and then the other one that I snuck in here is basically a comparison between ceftriaxone and penicillin for neurosyphilis. So this was done in 9 sites throughout Mexico, and it is retrospective. But they looked at people who were treated with penicillin, IV penicillin at our standard dosing, versus ceftriaxone at 1 or 2 grams per day, IV or IM. And this was, I think they said both were basically 10 to 14 days. So since it was retrospective, I guess it wasn't entirely standardized, but duration was the same for both. And then for the outcomes they looked at were interesting. They looked at the 4-fold greater reduction or greater reduction in RPR or VDRL, but they also looked at clinical response at 6 months, which was basically a clinical response included pleocytosis or the subjective report of symptom resolution. And that was, even though it's called early response, it was at 6 months. So what you can see here is that serologic response was technically more likely at 6 months in the ceftriaxone group, but I guess it's statistically significant, but 47% versus 42%. I don't know the clinical meaningfulness on that part, but they did show that the clinical response was actually worse in the ceftriaxone group. So with 28 versus 58%, and this is again at 6 months. Which I did find quite surprising. Overall, the groups were pretty well matched, although they do note that there was a significantly higher percentage of people with positive CSF VDRL in the ceftriaxone group, with like 74 versus 26%. But once they did multivariate regression, they didn't see any statistically significant differences in either clinical or serologic response. And I think it is interesting to note that 97% of the people with on penicillin were hospitalized versus only a quarter of those who had the ceftriaxone. And so and the other component was they did find that hepatitis B co-infection did have significantly lower odds of a clinical response as well. And so, their take-home message was basically that there was no significant difference found in early clinical or serologic responses between those who received penicillin versus ceftriaxone, but they did warn, which I think is very reasonable, to increased follow-up is certainly needed, or closer monitoring is definitely needed, but I'm not entirely sure I'm ready to do this yet, but there are, I think we've all seen some circumstances where we have to have alternatives, and this was somewhat reassuring in that way. Or sometimes we see the people who need to be on ceftriaxone for something else, and it's possible that we could avoid penicillin in addition. And then looking at just, I think another theme was sort of addressing heterogeneity. So, I've talked a little bit about it before here, and so has Scott and lots of other people, of trying to come up with better ways of classifying basically neuropsychiatric or behavioral or brain health disorders that don't rely so much on kind of a clinical syndromic type of definition, but more biologically based or mechanism based. And so this was an interesting study out of Boancis's group saying analyzing neuroanatomical heterogeneity in cognitive impairment. And so what they did was semi-supervised machine learning. They told it the features that they were interested in, but they were able to find 3 distinct neuroanatomical subtypes. And this was a group of 244 people without HIV and 366 with. And I think the most important and interesting part to me is if you look at the control participants versus people with HIV, just as 2 lump groups, you actually don't see significant differences in the imaging pattern. However, with the machine learning, you do see it actually breaks the people with HIV into 3 distinct clusters that are significantly different. And these are significantly different from people without HIV. And so this really, I think, highlights that the importance of looking at more heterogeneity and more finer resolution to aid who get clinical trials, targeted treatments, and things like that. And overall, the clusters did have some significant differences, and it wasn't entirely clear in their poster until I got to this part here, is that these were not people who were all virally suppressed. And so it's not too surprising that there was a cluster 3, which showed greatest atrophy and significantly higher viral load. So these are people whose HIV is not well controlled, and they do worst in cognitively and have the greatest atrophies and abnormalities on MRI. But there is cluster 2, which had low CD4 nadir count, but primarily were mostly undetectable, and most of them were on ART. And they also had, you can see differences here. So they actually had kind of an opposite pattern of kind of increased or hypertrophy in some areas of cortical function structures. And they still had pretty steep decline for psychomotor and processing speed. So I think that this was a very interesting study and really highlighted the value of the kind of looking at heterogeneity and classifications in a new way, which segues into my poster. And so this is kind of with the same concept overall, that we've had a lot of negative clinical trials for depression and cognitive impairment in people with HIV. And one argument about this is that it's likely that we're enrolling a very heterogeneous group with many different mechanisms, and so we're not really targeting enrollment or targeting treatments. And so this was trying to look at ways to basically adapt our classifications and ways to try to improve them, also using machine learning like the last group had done. And so I know this has been presented a few times, but just as a brief refresher, these are 4 neurobehavioral phenotypes that were previously identified in the charter group led by Dr. Letendre. And these are 4 different groups. Overall, NBP1 is healthy. The second one is mostly mostly depression, but other measures are okay. And then MBP3 is unhealthy across kind of all measures, this magenta one. And then 4 is predominantly cognitive impairment in blue, but other measures were okay. And so what we had done is we had measured a targeted proteomic inflammatory panel. And then they also compared to these neurobiological or neurobehavioral outcome measures. And overall, I know that I think I am running out of time, but I will just say that overall, what we found was if we look at these CSF protein profiles, they were significantly different. We measured them against several different outcome measures. And in the interest of time, I will kind of just say that there were 3 here. So CD40 and FLT3L and MMP10 were most commonly associated as significantly associated with many different outcome measures. So I think of them more as a kind of sensitive but non-specific measure. And these are primarily at kind of the innate and T cell interface. And then MMP10 has been associated with Alzheimer's disease in the past and increased neuroinflammation. And so just to take you through a couple ones that I think are most interesting would be that if you can remember, so MBP2 and 3 both have depression as a prominent feature of them. But if you look at their inflammatory profiles, they are significantly different. So MBP2 has significantly increased kind of true T cell activation markers of CD8A, CD5, whereas MBP3 is a cohort of people, or is a group of people that does have a lot more comorbidities. A lot more women are in this group as well. And this has many more associations with inflammatory proteins, but even kind of within the same cluster, it's more of the interferon gamma and TNF signaling pathways that are really elevated in this MBP3. And then, like I said, both of these have a prominent depression feature. If you look at Beck total or total depression scores, there actually weren't any of these associations, which I think really highlights that kind of trying to further refine our diagnostics and our classifications is really important for moving forward in designing basically enrollment criteria for clinical trials and getting targeted treatments going. And that is where I will stop. Hand it over.
Thank you, Katie. I think we leave questions till the end to make sure that we get all the information from Dr. Letendre. So I'll briefly introduce him. He's a professor in residence at the University of California, San Diego, with dual appointments in the Division of Infectious Disease and Global Public Health and in the Department of Psychiatry. Dr. Letendre performs translational patient-oriented research of the central nervous system complications of chronic infections like HIV and CMV. This includes observational cohort studies and interventional clinical trials that focus on the brain health effects of medical conditions and polypharmacy, the pharmacokinetics of antiretroviral drugs, and the biomarker correlates of disease. He has chaired the Neurology Collaborative Science Group at the AIDS Clinical Trials Group, the International Workshop on HIV and Aging, and the UCSD Institutional Biosafety Committee. He leads the NIH-funded multi-center Charter Plus cohort in the US and has participated in international research projects in China, India, Romania, Russia, South Africa, Zambia, and Mozambique. Dr. Letendre has mentored more than 75 trainees, has received a K24 Mentoring Award for the National Institute of Mental Health, and leads 2 NIH-funded training programs. He balances his research activities with clinical care of persons with HIV and other infectious diseases. He also teaches in the microbiology course at the UCSD School of Medicine, as well as a graduate course on research methods. Dr. Letendre has co-authored over 400 peer-reviewed manuscripts published in journals such as AIDS, Antimicrobial Agents and Chemotherapy, Brain, Clinical Infectious Diseases, Journal of Antimicrobial Chemotherapy, Journal of Infectious Diseases, Journal of Neurovirology, Journal of Virology, Lancet HIV, and Neurology. And we're looking forward to hearing a summary from the CROI adventures this year. Please take it away.
So, and we're out of time. I have to shorten that. Sorry about that, Anik. I'll make it shorter next time. Okay, so Katie did a great job. Those data on the O-link are particularly difficult to explain, and it's really nice to see how well Katie took us through that. So I'm going to go quickly also so we can get done. These are data from Ron Swanstrom's group at UNC, presented by Laura Kinzer, and it was a poster, but Laura also presented in a theme discussion session. And they had data on over 100 people, but these particular analyses focused on a subgroup of 45 who had untreated HIV. And you might be familiar with what they've done before, which is they used an assay developed decades ago by Ben-Hur Lee that they modified to identify the extent to which a virus must use CD4 in order to enter a cell. And that's relevant to the brain because there's not much CD4 produced there or expressed there. And so they look at genetic sequences to characterize people as having equilibrated or not compartmentalized HIV, and people who have compartmentalized HIV, meaning that the virus that they see is different in CSF than in blood. And then they use this assay to divide a CSF-derived virus into either M-tropic or T-tropic virus. And the M-tropic does not require CD4 to enter cells and is typically more avidly binds to CCR5. So they've been publishing on this for years, and it's valuable. But initially, I was struggling to understand what the incremental addition of these findings were to what they've shown already. But a couple of them are highlighted by Laura here. One of them is highlighted by Laura here, and I think it's valuable. And that is, we used to see, decades ago, in the early days, that people with cognitive problems, more severe like dementia, had higher HIV RNA levels in CSF than in blood, or than people who did not have cognitive problems, rather. And we don't see that anymore. And so they've used a very simple questionnaire to divide people into symptomatic or asymptomatic, and that's a little bit of a limitation here. But what you can see at the bottom right is that they saw that people who had M-tropic, or interestingly, I noticed yesterday, unknown tropism, had higher, symptomatic people with those types had higher HIV RNA levels in CSF than people who are asymptomatic by a lot. And the T-tropic virus or the equilibrated virus people didn't have those same relationships. And so, for me, frustrated over the years by seeing inconsistent results in things like viral loads and protein biomarkers and so on, this suggested that part of the reason we may see inconsistencies, one is that it may be our hypotheses are wrong. We always have to think about that. But another is that we're missing a critical piece of datum, and that critical piece could be tropism, that some of the associations with viral load here would only be seen in people who have M-tropic or this unknown tropism virus. And they went on to compare it to neopterin. Dr. Riggs mentioned NFL or neurofilament light, other proteins or proteins that we and others have seen in association with cognitive problems, that the relationship between viral load and those biomarkers was only present in people in the M-tropic group, not in the not compartmentalized or equilibrated group. So again, it was interesting to think about that when we're doing these studies, we're missing a critical piece of information about tropism that informs, that affects the relationships between cognition or brain health, measured by different metrics, and those biomarkers. So they sort of went through and reinforced. Dr. Kinser went through and reinforced again many of the findings that they've brought up before. So there's not very much new here, but I thought it was very intriguing, this idea that M-tropism, compartmentalization, is not surprising that it would affect the brain differently between individuals, but among the people with CSF compartmentalized virus, M-tropism further modifies it. And it makes sense that that might be true. I just hadn't thought of it in that way. So along the viral line, Mattia Trumfio and Sarah Janella and others, I see Cheryl Delano's here, and Dr. Riggs has contributed to the last GIFT program. Mattia showed some very nice data from the last GIFT program. Thanks to all of you who helped refer people with HIV to this important study, in which people have a life-threatening diagnosis, and when they before they die, they donate their tissues to the scientific study, and then when they die, Sarah and the team come in the middle of the night, usually, to collect the tissue. So a huge amount of dedication here, both from the participants and from the research team. And one thing that this focuses on is, in addition to looking at brain tissue, is looking at sort of the forgotten part of the central nervous system, the spinal cord. And in part, that's a bit because we don't see myelitis like we used to see back in the bad old days. But just as we see milder cognitive impairment than we used to see, and we see milder neuropathy, peripheral neuropathy, than we used to see, it's possible that we're overlooking the spinal cord effects because they're milder and we're not really attuned to it anymore because the myelitis was so terrible. So Mattia and the team were interested in the HIV RNA levels and DNA levels in the spinal cord and whether they differed or not. And there's more data presented than I'm going to show you in the interest of time, but they did show that in some instances, like in thoracic spinal cord, levels of HIV DNA were higher, although overall they were not. There's a lot of abbreviations here. I didn't take the time. I apologize to spell them all out. The SC down here at the bottom is spinal cord, and then the C is cervical, lumbar, or thoracic, pons, PBMCs, occipital lobe, and so on. So hippocampus, frontal, medial cortex, basal ganglia. So, but the individual findings aren't so important as, in this instance, the compartmentalization. So Mattia and the group found compartmentalization in the spinal cord distinct from other parts of the central nervous system, which I think is a really interesting finding. And this shows spinal cord versus brain, spinal cord versus blood, and brain versus blood for the percent of participants with compartmentalization. And so there are some differences, not only comparing the spinal cord to blood for compartmentalization, but to spinal cord and brain. So, one important consideration in all of this is when we're looking at CSF, as we do, and as Laura Kinser did with Ron Swanstrom, the data I was just showing you, where's that virus coming from within the central nervous system? And someone asked, so at the session, so do you think there are three compartments in the central nervous system? And, Laura, I think, answered it well, but essentially saying what I was thinking too, which is, well, there's more than one, right? So there's regional differences in the brain. there might be regional differences in the spinal cord. And so it's a complex environment. And yet we find important and interesting results. But this really supports the idea that the spinal cord, again, is sort of an overlooked area when it comes to HIV reservoir in particular. Now, this is a bit outside of my area of expertise. Mattia is also working with Sarah and the team on the T cell repertoire. And this highlights a very important area of the adaptive immune response that's been largely overlooked in the NeuroHIV area, the field. And Katie has been focusing on virus-specific epitopes for B cell responses, so for antibody immunoglobulin responses. Mattia's focusing here on the T cell repertoire. And just remember, there is an alpha and a beta receptor, and that these can vary, they do vary, depending on the antigens that the T cells interact with. And there's many different ways of characterizing this. Can folks see my cursor? I'm pointing over to the lower left. So the repertoire diversity, looking at the diversity of the T cells, and then when there are clonal expansions, for example, to particular epitopes of CMV or EBV or other herpes viruses, which we increasingly think are important in brain health and other aspects of health. So Mattia and the group can use various measures to look at this clonality and diversity. And this is just showing the alpha and beta receptors and showing that there are differences in HIV in the middle panel here. And on the right panel, looking at alpha and beta again, showing that the lower the repertoire diversity, the greater the activation. And on the bottom right, it's just one graphic that Mattia provided me that also looks at transition from mild cognitive impairment in people without HIV to Alzheimer's, showing that you can predict risk for transitioning to Alzheimer's from MCI using the T cell repertoire. So evidence here to support that it's important in people with HIV, or it's altered in people with HIV, and that it's important in brain health. And so there's various ways that scientists can use to characterize this, including sequencing, which is what we're going to show you here. Mattia and Sarah and others are also working on combining it with antigen specificity. But Michael Corley is here now from Columbia and or Cornell, sorry, New York people, sorry, Tim, from Cornell. And you can look at chromatin accessibility like Michael does, and gene protein expression as well. So this is a beautiful graphic, as Mattia tends to make, and combines a lot of data. But you can see, again, these regional, different regions of the brain represented here, along with thoracic spinal cord. And essentially what Mattia found, each of these little graphs that are here show HIV DNA, T cell repertoire richness, Shannon, D50, clonality. I can't explain the intricacies of all that these different metrics mean, but what Mattia found is more associations than you would expect, many more, based on chance alone. And Katie and I and others have found particularly that inflammation is associated with, which seems so broad and, amorphous compared to this level of analysis, but particularly associated with motor functioning. And what you can see here in the upper left is that in the frontal motor cortex, several of these metrics were associated with worse is to the right here, worse motor functioning in our performance in all of these T cell repertoire metrics. And so, again, in the interest of time, I won't take you through each of these. I can't explain why these are different. It's just very provocative and interesting. And the larger theme of of these newer technologies moving forward, analyses of the adaptive immune response like Mattia is doing and like Katie is working on, I think are very promising. All right, so what about therapeutics? So Dr. Wilkins tried to steal my thunder earlier, and that's not surprising because there was no neuro in this presentation. So Dr. Letendre, why would you present this? But Peter Hunt and Sarah Janella, again, who are leading this clinical trial of letermovir in the ACTG, were very gracious in including some neuro sub-aims. And so we're analyzing those data. And so, but the findings, even without those data, still have implications, not only for people with HIV who are in our clinic, but also for the nervous system. So you'll remember, I hope, that this is a anti-CMV specific antiviral. And in this study, Peter and Sarah and the team randomized people 1-to-1 to either letermovir along with their suppressive antiretroviral therapy or CMV for 48 weeks, and then followed them for an additional 12 weeks off drug. The ACTG required them to have a futility analysis, which of course is not a bad idea in general, but it was at week 8, and poor Peter and Sarah were forced to choose one biomarker. Soluble TNF receptors 2 is what they chose, but it ended up being, in retrospect, a problem. So Sarah really presented this beautiful idea about why the study was stopped, the trial was stopped, because they had, rather than having a decrease in soluble TNF receptors, they reported last year, or maybe it was longer than that, but that there was unexpected increase. So this trial was stopped for futility. And part of that, one fascinating thing about herpes viruses is that I think they all encode for either chemokine or chemokine receptor proteins that are highly homologous to human equivalents. And so in this case, CMV produces a viral, that's the little v there, viral IL-10, which has anti-inflammatory effects. And so in some ways, Sarah compared it to keeping the brakes on the immune response so that you don't have, you're not killing infected cells. But when you inhibit that, the viral IL-10 is stopped, and suppressing viral IL-10 is like taking the foot off the brake of the immune system. And so, instead of initially the soluble TNF receptors going down, they went up because the IL-10, the viral IL-10, was taken away. And so I thought, just something I really hadn't thought through, and I just think it's really fascinating, this idea of herpes virus encoded chemokines or immune proteins, something that interested me, almost 30 years ago when I started my career. And so it's just interesting to see it coming up again. So they saw a sustained reduction in IL-10 receptor activity over 48 weeks and reductions in several proteins associated with cardiovascular disease and cancer that they reported last year. And here's the finding that Tim referred to. So one of the several findings that Sarah presented was this change in CD4-CD8 ratio. And you see here, this is the 8-week finding. There was not a statistically significant difference between the arms at 8 weeks. And so another thing we didn't know about are the dynamics of this process, right? And so why does how long does it take this eventual change in CD4-CD8 ratio 40 weeks? How long does it take for that to emerge? How much inter-individual variation is there? All we can say is it's not 8 weeks, right? There's no difference at 8 weeks, but that's what the futility analysis was based on. So we saw these differences. Sarah saw differences between women and men, pretty marked differences. You can tell by the confidence interval here that there were not as many women as men in this, and also CD4 is less than 350 down here at the bottom. So I'm not showing you all the graphs, but here are a couple of more. But on the left are all the findings: the reduced CMV shedding, reduced CMV IgG. We previously found that higher CMV IgG was associated with worse cognition. Reduced plasma IL-1 beta, which is the graph here on the right, showing the change in IL-1 beta concentration. So pretty nice differences there. Eventually, the soluble TNF receptor did come down. Christine Erlandson's also working with Sarah and Peter and the team, and they looked at the 5 times chair rise time, which is a nice indicator of physical functioning. That improved in the letermovir arm, but not in the untreated arm. And then again, they showed reduced biomarkers that are causally associated with cardiovascular disease last year. So really nice findings with implications for the nervous system, but implications for a lot more. Now, the other thing I'm going to focus on is just semaglutide. So there were a lot of findings focusing on this at CROI. We're using GLP-1 receptor agonists a lot in clinic. We're very interested in studying it more, including for methamphetamine use disorder. But this is just a nice graphic that Michael Corley shared with me of the many biological and organ system effects of the GLP-1 receptor agonists. With respect to the brain, people in the field have previously shown that there can be insulin resistance compartmentalized in the brain, even when people don't have it outside the nervous system. And that's been reported with Alzheimer's as well. But there are other effects besides the effect on insulin resistance in the brain that GLP-1 and its receptor agonist can influence. And this is a nice meta-analysis that looked at many studies, including those relevant to brain health and addiction, and the nervous system reporting benefits. To the left of the dashed line is a benefit of GLP-1 receptor agonists. So again, there were a lot of reports at CROI this year. Many of them were based on Grace McCumsey's beautiful clinical trial that she organized and ran. Relatively small sample size, but statistically significant differences between the placebo and the semaglutide arm. Nonetheless, this on the right is just another nice graphic from Michael Corley, summarizing the different generations of epigenetic clocks. So many of the analyses, these are from 2 different presentations, as you can see down at the bottom left. But many of the presentations focused on biological aging as determined by, or as estimated by, epigenetic clocks using DNA methylation. And I don't remember always that the first generation compared DNA methylation algorithms to chronological age. Second generation compared them to mortality, and third generation, which are more recent, look at the pace of aging. So this is the abstract 172 that was presented in the oral neuro session. They use, this is from Dr. McCumsey's group, they used CogniView, which is a computerized neurocognitive assessment. Don't love it, but it's reasonable and a comprehensive assessment. And you can see the numbers here again, 108 people. And Dr. Athier, the presenter, nicely highlighted the differences here, that people who got semaglutide did better in delayed recall, naming and language, as well as visual spatial. And you can see visual spatial had the lowest p-value, and so the difference was greatest. And so perhaps not surprisingly, when they did multivariable adjustment, it was only the visual spatial score that retained a p-value less than 0.05. They did something very nice after that too, and they looked at mediation using multiple metrics that you can see defined down at the bottom to see whether or not the observed relationship between semaglutide and visual spatial scores, the strongest effect, was mediated by any of these factors that are listed up at the top: weight, visceral adiposity, high sensitivity CRP, soluble CD163 or soluble CD14. And what they found was that it did, the statistics supported that the effect of semaglutide on visual spatial performance was mediated by these indicators of inflammation and myeloid activation. So very nice analysis. Michael Corley's analyses also were presented in a separate session, focused again on biological aging. He found that semaglutide reduced epigenetic aging or epigenetic aging mortality risk by about 3 years and reduced the pace of aging by about 9%. So diffuse effects here, and here's just a list of the abstract number. This is not 113 abstracts, this is abstract number 113, but really the broad effects of semaglutide on multiple factors and associations of epigenetic clock measures with various measures, including cognition. So a few more clinically relevant findings, a couple more actually, just this one. This is from Crystal Wang, one of our trainees, something that is easily translatable to the clinic, calculating something called the Systemic Inflammation Index, or SII. It's just the neutrophil count times the platelet count divided by the lymphocyte count. Crystal analyzed data from about 2,000 people from our database and basically found and divided people into older or younger, which was arbitrarily defined as a youthful 50 years, and found that the SII was associated with worse motor performance, again, only in the older group, and was overall associated with poor global cognition. And Anik, this will be my last slide. So another one of our trainees, Azin Tavasoli, also used our database working with Ron Ellis. Both Crystal and Azin worked with Ron, and looked at the association between cognition and the number of antihypertensives. And it's just a reminder that we tend to think that people who have cognitive impairment are at greater risk for being challenged by adherence with their antiretrovirals, but it's not limited to antiretrovirals. So as you heard from Katie, there are still issues with polypharmacy, even though antiretrovirals don't contribute to the number of pills as much anymore as they once did. But what Azin found is that over time, again, in these a large number of people here, about 1,500 people, over time, poor cognition was associated with a larger number of antihypertensives. And she wasn't able to say, chicken, egg, both. Which came first? Is it that people have worse hypertension that's refractory to treatment, and that's associated with poor vascular outcomes and worse cognition? Or is it poor cognition leads to people forgetting to take their antihypertensives, so their blood pressure is still up when they come into clinic, and we add more and more. But just remember to think about this when you're seeing people in clinic. So lastly, this was not on the list of Katie's abstracts that she was going to present, so I put it in here too. So I won't cover it because she already did, but it reinforces that we both thought it was important because we both put it in here. So there's not much time left. It's 9 o'clock, but I'm happy to take questions, and I'm sure Katie is as well.
Thank you so much to both of you for taking the time to put together these beautiful summaries for everyone who didn't get a chance to go or who didn't get a chance to attend those sessions. I guess we'll close the session today, but you can contact Katie or Scott if you have any questions. I see Bobby.
Anik, can we just stay on for people who are still here, and if they want?
Yeah,
is that sure, okay?
For sure. Yeah.
So I'm willing to stay for a few minutes if folks want to ask questions.